In this lecture, we will discuss the degradative pathways of aromatic amino acids tryptophan, tyrosine, and phenylalanine. Let's first see the degradative pathway of tryptophan. Tryptophan, in the presence of tryptophan 2,3 dioxygenase, takes in the oxygen and forms N-formylkynurenine. N-formylkynurenine, in the presence of formamidase, takes in a water molecule and a formate ion is released out, thereby forming kynurenine. Kynurenine, in the presence of kynurenine 3-monooxygenase, takes in the oxygen and releases water molecule, and NADPH gets oxidized to NADP+, thereby forming 3-hydroxykynurenine. 3-hydroxykynurenine, in the presence of kynurenase, forms a skiff base, and the bond between the beta and gamma carbon is broken down, and 3-hydroxyanthranilate plus alanine is formed. In this process, a water molecule is taken in, and thus formed alanine molecule gets converted to pyruvate. 3-hydroxyanthranilate takes in the oxygen molecule, and in the presence of 3-hydroxyanthranilate 3,4 dioxygenase forms 2-amino 3-carboxymuconate 6-semialdehyde. This spontaneously gets converted to quinolinate, which acts as a precursor in the formation of NAD and NADP. On the other hand, 2-amino 3-carboxymuconate 6-semialdehyde, in the presence of amino carboxymuconate semialdehyde decarboxylase, forms 2-amino muconate 6-semialdehyde. Here a molecule of carbon dioxide is released out. This 2-amino muconate 6-semialdehyde, in the presence of amino muconate semialdehyde dehydrogenase, forms 2-amino muconate. Here a molecule of NAD plus gets reduced to NADH. 2-amino muconate, in the presence of hydratase enzyme and dehydrogenases, forms alpha ketoadipate. Alpha ketoadipate follows a series of reactions to form acetoacetate. The reactions involved in this are the same as that involved in the lysine degradation. We will discuss these steps in lysine degradation. Through these reactions, two molecules of carbon dioxide are released out and acetoacetate is formed. Thus, we can say that tryptophan is both ketogenic and glucogenic, because it releases both acetoacetate and pyruvate. Now let us see the degradative pathway of phenylalanine and tyrosine. Phenylalanine is initially converted to tyrosine, and both phenylalanine and tyrosine have the same pathway of degradation. Phenylalanine, in the presence of tetrahydrobiopterin plus oxygen, gives dihydrobiopterin and water in the presence of phenylalanine hydrolase, thereby forming tyrosine. Tyrosine, in the presence of tyrosine, in the presence of aminotransferase alpha ketoglutarate gets converted to glutamate, and para-hydroxyphenylpyruvate is formed. This p-hydroxyphenylpyruvate, in the presence of p-hydroxyphenylpyruvate dioxygenase, forms homogentisate. Here an ascorbate and oxygen molecules are taken in, and dihydroascorbate plus water and carbon dioxide are released out. Homogentisate, in the presence of homogentisate dioxygenase, takes in a molecule of oxygen and forms 4-mellyl acetoacetate. This 4-mellyl acetoacetate, in the presence of mellyl acetoacetate isomerase, forms 4-fumaryl acetoacetate. 4-fumaryl acetoacetate, in the presence of fumaryl acetoacetate, takes in a water molecule and forms both fumarate and acetoacetate. Thus, we can say that phenylalanine and tyrosine are also both ketogenic and glucogenic, as fumarate is the intermediate of TCA cycle and